Alright, welcome to Chem 313, Lecture 13. To review from last time, we talked about carbohydrates, and carbohydrates are an awful lot of terminology. So to review that subject, I'm going to say you should refer to the carbohydrate worksheet, which is up on Canvas. And then there are a couple of pretty lame carbohydrate questions on homework 6. But I'm going to de-emphasize pure carbohydrate questions in this course. So you're only responsible for easy questions on quizzes or exams regarding carbohydrates themselves. And they're not going to be worth a whole ton of points. What is going to be more important for this class is the fact that carbohydrates are used as fuels, which is a fancy way of saying food, in most organisms. So essentially all higher organisms, all eukaryotes, and the majority of bacteria use carbohydrates as fuels. This is a primary food source for us. But what we haven't had a chance to talk about yet is what exactly does this mean? So in today's lecture, we're going to introduce the metabolism section of the course, or biological energetics, which is going to deal with how we actually obtain energy from fuels, and a few other subjects as well. All right, this molecule, ATP, I think everybody knows that this is the major cellular energy source. But again, what exactly do we mean by that, right? We can say that, sugar is an energy source, right? Sugar has caloric value, it contains energy. So what do we mean when we say that ATP is our primary cellular energy source? What we mean is this. This reaction, which is hydrolysis of ATP, okay, ATP plus water react to give us ADP plus phosphate. This reaction is very favorable. And what do we mean by favorable? We mean that it has a delta G that is significantly negative. So the standard delta G for this reaction is about minus 31 kilojoules per mole. You don't need to know that number. Don't attach any significance to it. But under normal cellular conditions, it's actually lower than that. It's around minus 50 kilojoules per mole. So why is that? Well, you'll recall that for any chemical reaction, delta G is equal to delta G standard plus RT natural log of Q. Delta G standard is a constant that is the free energy change of the reaction under standard conditions, which is usually taken to be 0 Celsius, 1 atmosphere pressure, all solutions at 1 molar concentration. This second term here is a variable, and the reason it's a variable is primarily because of that Q there. That Q is what's called the reaction quotient. So the reaction quotient is equal to the product of all product concentrations at that given point in time, raised to their stoichiometric coefficients, divided by the product of all reactants concentrations at that moment in time, raised to their coefficients. So for example, the reaction quotient Q for the hydrolysis of ATP would be the product of all product concentrations, so ADP concentration times phosphate concentration divided by the concentration of ATP. And remember, we're leaving out water here because water is a liquid and pure liquids don't factor into reaction quotients. All right. So notice that if Q is small, right? If it's small, that means you've got lots and lots of reactant and very little product, and or very little product. That means our delta G is going to come out to be more negative. We're going to come back to that point later on, all right? But suffice to say for right now that delta G for ATP hydrolysis is going to be very negative. Why is that important to us? The reason that's important to us is because if we have any reaction that is unfavorable or non-spontaneous, that is, delta G is positive, it can be made favorable by coupling it to ATP hydrolysis. So for example, let's say that we have some reaction A breaks down into two molecules B plus C. And let's say that the delta G is positive 24 kilojoules per mole. Okay, so this reaction, because it has a positive delta G, it's going to be non-spontaneous. If you want to get it to work, you have to force it. But we said before, ATP plus water react to give us ADP plus phosphate. That has a delta G of about minus 50 kilojoules per mole. So we can combine them. If there's some way that you can make the reaction A forms B plus C happen at the same time more or less or in the same place as ATP hydrolysis, then our overall delta G is now going to be negative 26 kilojoules per mole. So we've made an unfavorable reaction favorable by coupling it to ATP hydrolysis. That's what we mean when we say that ATP is an energy source. It can be used to power unfavorable reactions. We have a problem though, and that is ATP has to be made. All right, so the adenine ring up here and the ribose ring, those are synthesized, but when I say ATP must be made, what I mean is that we have to simply reverse this reaction up above. ADP plus phosphate react to give us ATP and water. So cells don't just have an infinite supply of ATP lying around, they have to make it by running this reaction. But this reaction is the reverse of the reaction listed above, which means that its delta G is about positive 50 kilojoules per mole. So ATP is wonderful, we can use it to power unfavorable reactions, but how do we power the unfavorable reaction of making ATP itself? Right? In other words, if ATP is a high energy molecule, we have to get some energy in order to make it in the first place. And where is this energy going to come from? So that's the topic that we're going to be spending the next couple of lectures talking about, is where do we make our energy to power this synthesis of ATP? Because clearly energy must be put into this process if we're going to make it happen. All right, so that's our lead-in into the subject of metabolism. Metabolism is the rearrangement of matter and energy into useful forms in living cells or systems, since metabolism can be spread out among different types of cells in some organisms. Okay, so into useful forms we can have examples. So what are some examples of rearranging energy and matter? Let's talk about rearrangements of energy into useful forms. So we're talking about taking things like chemical energy from fuels and converting them into different forms of chemical energy. For example, chemical energy in the form of ATP. That's what we're going to emphasize in our talk about metabolism in this course. But it doesn't stop right there. Okay, we can also take energy in the form of light. That would be photosynthesis, which we don't have time to talk about in this course. And that would also be converted into chemical energy. In some organisms, heat can be directly converted into chemical energy. Notice that it's usually chemical energy that we're forming, but not necessarily. There are some organisms that produce light as part of their metabolism. As far as matter is concerned, we can do things like convert fuels to wastes. Now, wastes are not, useful by definition, but the process of breaking down fuels into wastes can give us useful side products. Another example would be to take organic building blocks and convert them into biomolecules. So these are examples of energy conversions and matter conversions that would fall under the realm of metabolism. So metabolism kind of divides itself into two classes of metabolic reactions. We have what's called anabolism, and what is anabolism? Anabolism is when you take smaller molecules, simpler molecules, and through a series of chemical reactions convert them into larger, more complex biomolecules. And typically, anabolism requires an input of energy. The other type of metabolism, which is what we're going to spend more time talking about, is catabolism. Catabolism is when fuel sources or other large molecules are broken down into small molecules and or wastes, and in the process energy is released as a side product, although usually energy is the point of doing catabolism in the first place. So in this course we are going to focus on catabolism in the next unit of the course. We will talk briefly about one particular anabolic pathway just to kind of complete the story of how carbohydrate metabolism works, which is sort of the central theme of this part of the course. But that's where we're going. We're going to emphasize catabolism, which is how the cells break down fuels to get energy, which is then used to make ATP. So in metabolism, there's a tendency for instructors to go overboard with instruction and for students to go overboard in trying to remember everything. So I want to make it clear up front what you need to understand from this unit of the course. First of all, general principles. Of how energy can be obtained from matter. What I mean by that will become more evident on the next slide. All right. For any biochemical pathways, or processes, you should know the major inputs and outputs. Okay, in other words, reactants and products. And by major, I mean you don't have to account for every last proton or every last water molecule, but the major carbon-containing compounds. You need to know the regulated steps and how they're regulated. You need to know how and why regulating one process might affect a different process. So, for example, inhibiting one reaction might cause the reactant from that reaction to undergo a different reaction. That sort of thing you have to be aware of. Next thing that we need to know is an understanding of reversibility. Okay, what is it about reversibility? So we need to define that term in a moment here. But before I do that, I'm going to give you the last thing that you need to understand, and that is the overall point of each process. All right, what's it trying to do? What's it supposed to accomplish, to put it more precisely? All right, so reversibility, this has to do with free energy. So one thing I'm going to be saying repeatedly in this class is that some reactions can go in reverse and some cannot. And that is a surprisingly important fact in metabolism. You have to keep track of the fact that some reactions can go one direction and some can go in both directions. So why do we care about this? That'll become evident later on. But what is reversibility? What makes a reaction reversible? So I'm not talking reversibility in the strict thermodynamic sense. I have my own definition of reversibility. A reversible reaction, for the purposes of this course, means that its free energy is near zero. All right, well, why would that be a reversible reaction? Well, remember from Gen Chem, if the delta G is equal to zero, the reaction is at equilibrium. So if the delta G is near zero, that means the reaction is near equilibrium. And if it's near equilibrium, then a buildup of products, say, right, might end up pushing the reaction the other way, right? So this reaction can reverse if the concentration of products builds up or the concentration of reactants drops. On homework 6, there is a problem related to this, and I encourage you to do that problem to understand what I mean by reversible and irreversible. An irreversible reaction has delta G significantly below 0. And so the cutoff that I use, and it's arbitrary, but my cutoff is minus 10 kilojoules per mole or less. Not that you have to know the delta G's of any reaction. That's ridiculously fastidious. Okay? That's just where I draw the line, minus 10 kilojoules per mole. These are reactions that are far from equilibrium. Okay, trying to reverse a reaction that is irreversible would be like trying to get Niagara Falls to flow uphill backwards into Lake Erie by building a dam downstream. You'd have to build one very, very high dam to get the water to back up enough that Niagara Falls would flow in reverse. And this is the same sort of deal. The reaction is so favorable that there's essentially no amount of product buildup or reactant depletion that would make it flow in reverse. And as we're going to see, irreversible reactions tend to be very important reactions. One thing you are not required to do in this class is memorize all pathways. You're welcome to try, but I am not going to have you write out the name and structure of all 10 intermediates of glycolysis. I think that's a waste of time. However, knowing your pathways, the more you know about them, the easier it will be. But you're not going to be specifically tested on memorizing anything for this part of the course. All right, we are going to focus our attention on one form of catabolism. So that is the major form of catabolism on Earth. And that is what is called aerobic respiration. What's aerobic respiration? It's the process of generating energy for ATP synthesis by oxidizing carbon-rich fuels and in the process reducing oxygen. So aerobic respiration works like this. We take our fuels, which are our food sources, and we oxidize them. Our oxidizing agent is O2 gas, which is abundant on Earth. And then what we do is we burn them essentially until we have CO2 and energy. And that energy can then be used to power ATP synthesis. All right, so it's a little bit more complicated than I showed it right here. So we do not literally mix fuels and oxygen and react them to give us CO2 and energy. If we were to do that directly, what we would have would be combustion, which is what you get if you take, a slice of bread and set it on fire. I mean, yes, you are releasing energy, but most of the energy from combustion comes out as heat. And heat is the lowest form of energy. It's difficult to use. All it's good for is raising the temperature of things. So we don't do this directly. Aerobic respiration is a bit more subtle than that. Okay? The way it works is we break it apart into multiple stages. So typically the way aerobic respiration works is like this. We take our fuels and through a series of oxidation reactions, we generate CO2, which is a waste product. Now, oxidation reactions, what's oxidation mean? Oxidation means that you're losing electrons. So that means that we're going to take electrons from our carbon-rich molecules, and we're going to put them on molecules called electron carriers. You'll recall that if you add an electron to something, it becomes reduced. Right? That means that we need oxidized electron carriers as a reactant. All right, so what we do is we oxidize our fuels, giving us CO2. We pass off electrons onto electron carriers, and then what do? What happens next is, again, in a series of reactions, oxygen picks up the electrons, and it becomes reduced, giving us water. You have to have protons there to balance it out because water contains 2 hydrogen. This process is very energetically favorable, and it releases energy that can be used to power ATP synthesis. So this is the flow chart for how aerobic respiration works. We oxidize fuels to CO2, hand the electrons off to electron carriers. The electron carriers hand the electrons off to oxygen. The reduction of oxygen is very favorable. Okay? This is entirely possible because the reduction of oxygen is very favorable. Its free energy change is very negative. Why is that? Well, that's partially because oxygen loves electrons, right? It's up there in the upper right-hand corner of the periodic table, so that's where all the electron sucking up elements live. And also, oxygen is abundant on this planet, and that means our concentration of reactants is high, and that pushes the delta G into negative territory, right? Because the reaction quotient Q is products divided by reactants. If you've got lots of reactants, the reaction tends to be pushed forward. Now, there are some organisms that undergo anaerobic respiration, and those organisms live in environments where there's no oxygen. But the idea is essentially the same. We have reduction and oxidation reactions that are driven by some ultimate electron donor or acceptor picking up electrons, being favorable. This is just a convenient graphical depiction of what I already talked about before, which is that oxidation of carbon and reduction of oxygen are separate steps. And they're joined by electron carriers. It's almost like an electrochemistry reaction. You have your oxidation half reaction taking place in one cell and your reduction half reaction taking place in a different cell, and you've got a wire connecting them. It's very similar for aerobic respiration. Carbon oxidation takes place in one area, in one set of reaction. Reduction of oxygen takes place in a different set of reactions. The link between the two is the electron carriers. However, the energy of this process comes from the oxidation of carbon. All right, so that means that, recall that oxidation is electron loss. So any compound that has more electrons to lose is going to be a higher energy fuel source. So there's a rule of thumb, all right? When carbon is oxidized, oxidation is loss of electrons. When carbon is oxidized, it either loses bonds to hydrogen or gains bonds to oxygen. That's worth remembering. And conversely, when carbon is reduced, it either gains bonds to hydrogen or loses bonds to oxygen. Remember, reduction is gain of electrons. So as a result of this, compounds with more carbon to hydrogen bonds tend to be more energetic. And we'll see examples of that throughout this part of the course. All right, let's take a look at electron carriers. So once again, we said that the oxidation of carbon and the reduction of oxygen are two separate processes, and they're connected by electron carriers. We first oxidize carbon and reduce an electron carrier, and then later reoxidize the electron carrier and reduce oxygen. So what are these electron carriers and what are we talking about? There's two major electron carriers for catabolism. There's more than two in total, but there's two big ones that I care that you know about. Those two big ones are NAD plus and NADH. NAD plus is the oxidized version and NADH is the reduced version. This diagram shows you the structure of those molecules. You don't have to know the structure. It's big and complicated. This highlighted part is the business end of the molecule. What ends up happening is NAD plus picks up a hydride anion from a carbon molecule, and that generates NADH. Okay, so notice that a hydride contains two electrons. So NAD plus and NADH is a 2-electron carrier. You carry 2 at a time. But it doesn't just carry bare electrons, it carries a hydride. Okay, so notice that you start off with one hydrogen atom on that carbon at the top end of the molecule, and then you pick up a second one, and the positive charge goes away. So NAD plus and NADH, this is the major catabolic electron carrier. This process of carrying the electrons is energetically favorable. It's favorable in the oxidation direction, and this molecule carries enough energy to synthesize about 3 ATP, depending on some situations. The other major catabolic electron carrier is FAD and FADH2. FAD is the oxidized version, FADH2 is the reduced version. So once again, this is a big complicated molecule. Just like NADH, it's got an adenine nucleotide, and then it's got a linker, and then it has this flavin ring at the top, and that's where the electrons get picked up. What happens is FAD picks up a hydride and a proton to form FADH2. So once again, we're picking up a hydride, so FADH2 is also a 2-electron carrier. But it's less energetic than NAD and NADH. So it only has enough energy for about 2 ATP, okay? And when I say it has energy for 2 ATP, by the way, what I mean is that the energy is released when these molecules are oxidized, when they hand off their electrons to oxygen. There's another electron carrier that I'm not going to show here, and that's the major anabolic electron carrier. We're not going to have a chance to see it in this course, but I wanted to mention it. That is NADP plus and NADPH. NADP is the oxidized version, NADPH is the reduced version. So the function of NADP and NADPH is these carry electrons for synthesis of carbon compounds. So what happens in those cases is our fuels become oxidized to CO2, NADP plus becomes reduced to NADPH, but instead of giving up its electrons to oxygen, it gives its molecules up to oxidized carbon compounds, forming reduced carbon compounds, which can then be used to make biomolecules. So we're not going to see NADP or NADPH that much in this course. I might mention them in passing. The difference between NADH and NADPH is that NADPH, the P stands for phosphate. NADPH has a phosphate right there on the ring. That's the only difference between those and NADH. So I leave it as an exercise to you to figure out why would putting a phosphate on this end of the molecule, all the way down here on the opposite side from where the electrons get picked up, how would that target one of these electron carriers for catabolic processes and another one for anabolic processes? How would that work out, do you suppose? That's a hypothetical question to test your understanding of biochem. All right, so what we're doing here is giving you guys an overview of aerobic respiration. So this is how the process of fuels being oxidized ultimately results in ATP synthesis. All right, so what we have at the top of this diagram are our major fuel sources: lipids, carbohydrates, proteins. These are your foods, right? And so what ends up happening, you'll notice, is that for all of our major fuel sources, they get broken down into simple building blocks. So proteins get broken up into amino acids by the digestive tract. Carbohydrates get broken up mostly into glucose, but also other carbohydrates, monosaccharides as well. Lipids, the most important dietary lipids, contain fatty acids, and so those are made into fatty acids. Now at this point, the building blocks have to be made into some sort of metabolite that is easily oxidized. And the most important metabolite is this molecule, acetyl coenzyme A. I'm not going to draw the whole molecule, but it's an acetyl group, which is that, connected to a sulfur, connected to a gigantic, huge chunk of carbon and bulk that I'm just going to abbreviate as CoA. So this is a 2-carbon molecule for all intents and purposes. So our goal is to get all our different fuels into this substance known as acetyl-CoA. Okay, so the roles of the different pathways kind of depend on what the starting material is for getting your acetyl-CoA. So the pathway for going from glucose to acetyl coenzyme A is known as glycolysis. There's also an additional step called pyruvate dehydrogenase, which is usually included with glycolysis in most textbooks. All right. At that point, once we have our acetyl coenzyme A, we can then oxidize it, and the oxidation of acetyl coenzyme A takes place in a pathway called the citric acid cycle, also called the Krebs cycle. And so the goal of that cycle is to liberate CO2. We're going to talk about that process. At the end of this process, we've completed all of our oxidation of carbon, so we have electron carriers. Now, after we have electron carriers, the next phase of this whole thing is to reduce oxygen. And so that's the responsibility of what's called the electron transport chain. And that liberates energy to power ATP synthesis, which is at the very bottom of the slide. So we're going to talk about all of these pathways in this unit of the course. We're also going to spend some time talking about how fatty acids are made into acetyl-CoA. That's a process called beta oxidation. We're not going to talk about how amino acids from proteins are made into acetyl-CoA or used as other fuels. It's complicated, it's different for every amino acid, and to do it properly would just take up too much class time, so I'm not going to waste my time with it. All right. So notice that all of this stuff is the oxidation of carbon phase, and the bottom stuff is the reduction of O2 to liberate energy. And we're going to go through all of this stuff over the next couple of lectures. All right, here's our first biochemical pathway, glycolysis. Glycolysis is the process of taking glucose and getting it ready to convert to acetyl coenzyme A. And the key product of that is pyruvate. Okay, pyruvate is not acetyl coenzyme A, but it's converted into acetyl coenzyme A in a subsequent step. All right, so for all pathways in this course, you should know, this is true not just for glycolysis, but for all pathways we're going to talk about, you should know where and when it happens. All right, by when I mean under what biological condition. You need to know the major reactants and products. You need to know the regulated steps and how it's regulated. And then lastly, you need to know the big picture stuff. What is the point? What is the goal? All right, so that's what you need to know about glycolysis. So glycolysis, where and when does it happen? It happens in the cytoplasm of all cells, and when it happens is when ATP is scarce. All right, major reactants and products, those are given on the slide right here. Glucose plus 2 ADP plus 2 phosphate. Pi is shorthand for phosphate ion, plus 2 NAD plus react to give us 2 pyruvate, 2 ATP, and 2 NADH. Those are the major reactants and products of glycolysis. The regulated steps and how it's regulated we'll get to in a moment. The point or the goal. There are two goals. One, get a small amount of ATP without the need for oxygen. Oxygen is not involved in glycolysis, and it's not necessary for glycolysis. That's going to be important for reasons that we're going to see later. But assuming oxygen is present, the more important goal for glycolysis is to generate pyruvate, which can be used to make acetyl-CoA and further energy, or it can be used as a biosynthetic precursor of various other molecules. So glycolysis is 10 reactions in total. And I'm not going to expect you to rattle off all 10 reactions. They're all catalyzed by enzymes, as is normally the case. But we have two phases of glycolysis. We have the energy investment phase, which is the first 5 reactions. And it's called that because it actually consumes ATP. That may seem counterintuitive, but the second phase is the energy payout phase, where we get a net of 2 ATP per glucose. So in glycolysis, what we're doing is we're going to take a molecule of glucose, which is this, and eventually we're going to get 2 molecules of pyruvate, which is that. So it's an acid with an alpha keto group on it. So this is a 3-carbon molecule, glucose is a 6-carbon molecule, that's why 1 glucose gives you 2 pyruvates. So you've got 10 reactions, you're not responsible for all 10 reactions, you are responsible for the regulated steps. The regulated steps are step 1, which is catalyzed by an enzyme known as hexokinase, and that is the reaction that glucose forms glucose 6-phosphate, and it consumes ATP. That's this reaction right here. The next regulated step is reaction 3, which is catalyzed by phosphofructokinase. And phosphofructokinase reaction takes fructose 6-phosphate and converts it into fructose 1,6-bisphosphate, also consuming ATP in the process. So I told you that there's 2 ATP consumed, and that's why this is called the energy investment phase. So that's reaction 1, that's reaction 3. And the third regulated step is step 10, catalyzed by pyruvate kinase. That's when you take this molecule called PEP and get pyruvate, and in the process ADP gets converted to ATP. So how are these reactions regulated? All right, reaction 1 is inhibited by glucose 6-phosphate, its own product. This is an example of what's called product inhibition. Product inhibition is when the product of a reaction is a competitive inhibitor of the enzyme that catalyzes that reaction. So glucose 6-phosphate binds to the active site of hexokinase and prevents glucose from binding, right? Now, why would evolution have selected for an enzyme that is regulated in this way? Well, it's a way of feedback inhibition. If we get an excess of glucose 6-phosphate, that must mean that we're not using the glucose 6-phosphate we have. So it's a waste to convert more, especially since we have to consume ATP to do it. So it makes sense that this reaction would be inhibited by its own product. Reaction 3, phosphofructokinase, is inhibited by ATP and activated by a molecule called fructose 2,6-bisphosphate. All right, so what's the logic here? First of all, this is allosteric, both the activation and the inhibition. Okay, ATP inhibits. Why do you suppose that is? Well, again, let's look at the logic here. The entire point of glycolysis is to generate ATP. If we have a ton of ATP floating around, that must mean that we don't need to make more ATP. So why would we waste our ATP making more ATP, right? ATP is an inhibitor. My symbol for inhibitor is, I draw it in red with a negative sign next to it, right? That means inhibitor. Fructose 2,6-bisphosphate is an activator. I'm not going to get into where fructose 2,6-bisphosphate comes from. It's a molecule that is synthesized only to allosterically activate phosphofructokinase. Okay? The textbook talks a bit more about fructose 2,6-bisphosphate, and I suggest that you read it. This is the most important regulated step, the phosphofructokinase step. The reason it's the most important step is that it's the rate-limiting step for glycolysis. So this is the slowest reaction, and therefore it sets the pace for the entire sequence of reactions. So because it's the rate-limiting step, it's going to go the slowest, and if you choke off this reaction, you're going to choke off all of glycolysis. It's also what's called a committed step. What's a committed step? A committed step means that everything that comes after it is useless except for completing the pathway. So fructose 6-phosphate and glucose 6-phosphate, those can be used for other things. But molecules like 1,3-bisphosphoglycerate are not useful for other things. Once you have made fructose 1,6-bisphosphate, everything after that is only used for one thing, and that is to make pyruvate. So at this point, you are committed. Step 10, pyruvate kinase. It is inhibited by acetyl-CoA, and it is activated by fructose 1,6-bisphosphate. So fructose 1,6-bisphosphate is the product of reaction 3. So it actually activates the last step of the reaction. So think about it. That kind of makes sense, doesn't it? Because this is our committed step. We said everything that comes after that is useless except to make more pyruvate. So once we have fructose 1,6-bisphosphate, why not activate the last step so that we make sure that we are using up what we've already formed? Otherwise, we're going to be forming a bunch of useless intermediate. Okay? Acetyl coenzyme A inhibits pyruvate kinase. The logic for that is straightforward. What we're going to do with our pyruvate is make acetyl-CoA. If we've got a lot of acetyl-CoA, there's no point in making more. So, as with reaction 3, both of these are allosteric regulators. All right, so make sure that you can understand the logic behind these modes of inhibition, know the steps, know the names of the enzymes, but beyond that, don't drive yourself crazy trying to memorize glycolysis. I don't think it's worth it. In the next lecture, we're going to talk about what happens to the pyruvate after it's been synthesized. So I refer you to that lecture, and in the meantime, check out homework 6.